I'm going to be talking today about detecting structural variation in cancer genomes with long-read whole genome sequencing. So the landscape of structural variation in cancer, as you can see here from the Peacock 2020 study, this was all with short reads. But you can see that they were able to categorize quite a few types of structural variation, but there's still a lot of complex and unclassified structural variants that we see in cancer, and particularly in liposarcomas, osteosarcomas, and glioblastomas. We see that these classes are enriched. So for my project, I use sarcoma and glioblastoma samples. Just for a bit of context, sarcomas are a cancer of the bone and soft tissue. It's a largely understudied form of cancer, and it's quite rare, only accounting for 1.3% of UK cancer diagnoses. It's quite heterogeneous with over 50 different subtypes. And there's been little change in treatment outcomes since the 1980s, with the standard method being chemotherapy to shrink the tumor, followed by tumor resection. Fortunately, not all patients respond to chemotherapy, so they go through that unnecessarily. And from a genomic standpoint, sarcomas are enriched in diverse and complex structural variants, as I mentioned. Glioblastomas, on the other hand, are the most common type of brain tumor. They're quite aggressive and treatment resistant, with a 5-year survival rate of 6.8%. And from a genomic standpoint, they're also enriched in complex structural variants, and they frequently have amplifications via extrachromosomal circular DNA elements, which you can see on the right here, which is where an oncogene gets excised from the genome and then massively amplified. OK, so we've got some great clinical collaborators and also some collaborators at Genomics England, that'd be the labs of Adrian Flanagan and Greg Elgar. And with them, we've sequenced a range of adult soft tissue and also some brain samples from Lisbon. So we've got tumor and normal blood samples for each patient. So this is a quick snapshot of our cohort. We also have paired Illumina data for all of these samples. With the help of a postdoc in the lab, Jose Vela Inclin, we've aligned all of this data to Human Genome 38 with Minimap and BWA-MEM, and we've processed the Illumina samples with our in-house whole genome sequencing pipeline, which uses grids and purple to identify somatic, structural variants, and copy number aberrations. Of course, once we got to this point, once we had all the Illumina data analyzed, we wanted to analyze the Nanopore data as well. But at the point of my PhD, there weren't really any tools available to do this. So for my project of my PhD, I've been developing SAVANA, which is a somatic structural variant caller for long-read data. It's designed only for cancer samples, so it requires a tumor and a paired normal. And this would be a typical workflow of a project using SAVANA. So you take a tumor sample and a normal sample from the patient, perform long-read sequencing on each of those samples, align those samples to the genome, and then input the tumor and the normal BAM to SAVANA. We use the cigar string and supplementary alignment to identify potential breakpoints, and then cluster together potential breakpoints, both with the tumor and the normal ones being clustered together. What differentiates SAVANA is we then use a machine learning model to filter out noise and then classify variants into somatic and germline. The beta version of SAVANA is available on GitHub right now, which uses a decision tree classifier to do this categorization. But version 1, which is coming out in a couple of weeks, is using a random forest classifier, which we've observed gives improved results, and that should be coming out shortly. And for the rest of my talk, I'm going to be talking about the results from this random forest classifier. Yeah. OK, so how do we benchmark? I think we've heard mentioned before about COLO 829. This is a multi-platform reference for somatic structural variation. And it was sequenced with Illumina, Oxford Nanopore, PacBio, and 10x linked reads, which provides a set of 60 high-quality validated somatic variants. That's 118 breakpoints with a range of different SV types. But as I noted on this slide, the Oxford Nanopore data was sequenced with R9 technology, and it was sequenced and base called in 2017. So as we all know, there's been a large jump in the accuracy of nanopore calling in this time. So with our friends at Genomics England, we resequenced COLO 829 using R10 flow cells, and we've got the following coverages. So we were able to perform benchmarking on this newer version of COLO 829 data. And you can see this is the benchmarking on this resequenced COLO 829. This is using all the default and recommended parameters for these tools. And we can see that SAVANA, we've gotten the best precision, recall, and F measure on this data. Of course, though, we want to see whether we can do this well on Illumina samples as well. So we looked at how many somatic structural variants we captured that were called by the Illumina pipelines. Obviously, it's a bit of a challenge to say what is true when we don't have a complete picture of the genome with Illumina, but we figured as a baseline, we want to be able to identify the variants that are being picked up with Illumina. And we get an average of around 84% recovery rate with our current pipeline. OK, and then just to visually kind of show what this means is, so this is a recon plot. If you're interested in producing these, it's available as an R package on bioRxives from my supervisor and a postdoc in the lab. It's a great tool. Check it out. But we have here the total copy number in the black horizontal line and the minor copy number here. This is on chromosome 16. This is all from Illumina data. And we can see the vertical lines represent the structural variants as called by this pipeline. And we can see this is the result from SAVANA on this same sample and chromosome. We use the copy number because it gives a good proxy for a structural variant that is associated with a change in copy number from the Illumina data likely means that it's a true structural variant. So it serves as kind of a good proxy for being able to pick those up. And just as a note, the Illumina caller has access to this copy number information, but SAVANA and the rest of the callers, which I'm going to show later, they do not have access to this. So that's an added bonus that we're able to line up with those calls so well. Okay, and then when things get a little bit more complicated, we still do match very well with the Illumina data. And this holds true even when we have intense chromothriptic cases like the one that you see here. So that's really great to see. We also perform this comparison with the other colors. So you can see here we've got Illumina on the top left, SAVANA, Nanomon, and then we've got Swim, and Sniffles on the right. Just a side note on the right-hand side, those are not paired design. So we've been doing subtraction, and maybe we'll have to talk to Lewis about the best way to do that. But this is just one example of what the comparison looks like between the Illumina and the long-read colors. Okay. And then we also see here, this is involving a rearrangement with CDK4 and MDM2. And those are quite important cancer genes. It was reassuring to see also that on the right-hand side, where we see a flat copy number, we're not calling many structural variants. Yes, I'll just go through a couple more examples. So again, this is Illumina, SAVANA, Nanomon. This is on chromosome 4. And finally, this is a highly upregulated area on chromosome 3. And we can see that SAVANA recaptures those structural variants quite well, while being pretty flat in the rest of the chromosome. OK, so how to use SAVANA? I hope I've convinced you that it's a great tool to use if you're doing cancer genomics. The beta version is available on GitHub. If you'd like to use it, please test it out. You can also install it via Conda. The version 1 release should be coming in a couple of weeks. But in the meantime, if you have any issues installing it or any questions or comments about it, please feel free to reach out. I'm really happy to hear feedback about it at this stage. All right, and in summary, I'd just like to thank everyone in my lab, Adrian Flanagan's labs at UCL, and Greg Elgar's at Genomics England, and of course to all patient participants, and Nanopore for inviting me today. Thanks so much.